NEB TV, what's trending in science. Welcome to NEB TV. Today I'm joined by Eileen DiMolanta, who is a group leader for the NEB Next product development team. Hi Eileen.
Hi Dina.
And today we are talking about next generation sequencing.
First I'll talk about library preparation, and I'll be trying to do that in 60 seconds. Next, we'll meet members of the product development team to talk about a new NEB Next product that offers several advantages over other methods. Lastly, we'll hear from Kevin McKernan, the CSO of Cortigen and founder of Medicinal Genomics. He'll give his perspective on the future of next generation sequencing.
Great, let's get started.
In order to sequence a sample using next-generation sequencing, you need to make a library. That library consists of fragments of the DNA you are interested in, attached to short adapters at each end required for the downstream sequencing process. For library prep for Illumina, the first step is to fragment the DNA, repair the ends by filling in or chewing back overhangs, and phosphorylate the 5' ends. DA tailing then adds an A to the 3' ends of the blunted fragments, and adapters with a T overhang are then ligated. The final step is PCR amplification, which increases the amount of library and enriches for fragments that have an adapter ligated to both ends. To produce high-quality sequence data, you need a high-quality library that has equal representation of every part of the original DNA without any bias. A high-quality library will result in sequence data with uniform GC coverage, a low percentage of duplicate reads and chimeras, and a high percentage of reads that map to the reference sequence with no dropouts.
I'm here with Lina Pone and Ping Liu, two scientists who led the development of the NEB Next Ultra 2 products. Hey ladies.
Hi Dina.
So what were your goals for developing the new NEB Next Ultra 2 product line?
So we get a lot of requests from customers for the past 1 and 2 years saying that they have very limited sample and then they just have a difficult time making library. So we decide we just going to develop a new kit. We get very high quality of library from as low as 500 picogram of the human genomic DNA. But if you have that low complex genome, that you can go much lower than that.
I think it just allows researchers to use more samples. So those samples, like you say, where they just couldn't make a library, they now can make libraries. And ChIP-seq is one of those areas, those kind of unique protocols where often there's just not much library. And so this allows those researchers to use samples that they just couldn't use before. Most people think of it as occurring in PCR, where specific sequences are amplified in preference to others. And so what happens is you can end up with a library that doesn't completely represent your starting input. And so we spent a lot of effort limiting that bias so that the final product mimics your input. One of the biggest challenges I think we find in next-gen sequencing is it's changing. What people look at it changing. The metrics are changing constantly. And so what we want to do is when we're evaluating our kits and our products is to try to understand how our customers are using them and evaluating them. So trying to understand the metrics we need to use to evaluate the kits properly, whether it's sequence coverage, uniformity of coverage, duplication rates, chimeras. We need to understand best how our customers are looking at the products and using them. Yeah, I think low input, definitely as low as we can go. At one point, I think I made libraries from 5 picograms of human DNA. So now you're starting to open the door really to new technologies and new uses for next-gen sequencing in diagnostics and treatment and those areas which are so exciting.
Well, thanks so much for joining us today, and we hope that you'll be back soon.
Thank you.
Thank you.
Hi, I'm Kevin McKernan from Cortigen Life Sciences. We do a lot of clinical sequencing work in pediatric neurology. Currently, there's a lot of short-read next-gen sequencing going on. It's great for sequencing small gene panels to mid-size exomes and even whole genome sequencing. I think we're seeing most traction in the clinic in sort of the 1,000 gene area with exomes coming on board, but there's also a lot of unmet needs. we need to be able to pick up chromosomal microarray data in sequencing, and I think that's going to happen in the next year. Perhaps a year or two after that, I think we'll start to see some of the single molecule sequencing techniques perhaps capture the triplet repeats. If we can do that in next-gen platforms, that'd be even better, but we suspect some of these triplet repeats being very long in nature may require some single molecule sequencing that can capture those. But we're really excited because we can see many of these techniques all unifying on a single platform.
Eileen, thanks for joining us today.
You're very welcome.
And thank you for watching. If you have any suggestions for future episodes, please let us know.